Well, thank you very much. It's thanks for the invitation, and it's a pleasure to be here. And well, thanks to Jack for the kind words of introduction. Um, I mean, I guess what I'll do is I'll give so I'll start with a fairly broad overview of the field of aging and what we know and we don't know about the process of aging, and then I'll briefly mention some of the topics of research that we've been focusing on. Um, without going into a lot of detail, most of this has been published. I mean, I guess the point is, I'm sure you're aware that since the beginning of time, people have sought eternal life and youth. So there's been a number of, mostly men really, who've sought the fountain of youth. So I guess the first example I could find was Qin Shi Huang was a Chinese emperor who tried to achieve immortality by taking pills prepared by the court physician of the time, which unfortunately for him were mostly made of mercury. So actually, ironically, died of mercury poisoning. I guess the first scientific approach to immortality and to reversing aging was a Russian doctor called Bogdanov, who in St. Petersburg, his idea, which actually has some rationale now, was that young blood would have rejuvenating properties, which we now know to some degree that is actually true. There's all these experiments of parabiosis suggesting that. I mean, unfortunately for him, also he didn't know about things like different blood types, and he died after receiving a transfusion from a student who also had tuberculosis. So we're not entirely sure why he died, but it was certainly because of the blood transfusion. But it is the first scientific approach to immortality, the first scientific rationale, and he had a whole theory behind it. So that could be, I think that's really the start of this quest for immortality or for eternal youth. And what is really the advances in recent years have showed is that we now have the technologies, we now have the tools that, well, maybe they don't allow immortality or eternal youth, at least not yet, but at the very least, they will allow us to retard the process of aging and extend our human health and life. So let me start with a brief introduction. I guess I'll start just by defining the process of aging, which I'm sure you're aware of, but it's good to remember that, okay, so it is quite a striking process, as you can see in these Czech centenarians, but it involves various molecular, physiological, and cellular facets. So I like the definition of aging as a progressive deterioration of physiological function, accompanied by an increase in vulnerability and mortality with age. That's quite a broad definition that encompasses, I think, all of the different aspects of aging, but it emphasizes the decline or the deterioration of function, the increase in vulnerability, and the increase in mortality. So, for example, the chances of dying once you reach age 30 double roughly every 8 years. So that is the definition of aging, and then, as I'm sure you're aware, we're doing this from home. I mean, there's been this pandemic. That means we are locked down. I mean, as you might have seen on the news in Liverpool region, where I am at the moment, there's this tier 3, which I'm not entirely sure what it means, but it probably means I'll be staying home for quite a lot of time. And this is because of COVID-19, which of course is an age-related disease. I mean, this is data from China, but it could be from any country. As you can see here, the mortality of COVID-19 is much, much higher at advancing ages. So before roughly age 50, mortality from COVID-19 is relatively low. I mean, children can be affected, but unless they have underlying conditions, are rarely in serious danger. However, after about age 50, there's this exponential increase in mortality from COVID-19. So it's clearly derived because of aging. It is an infectious disease, but the reason it is a problem now, the reason we have this pandemic and lockdowns and disruption is because it is an age-related disease. there is an aging process that makes us more susceptible to this disease much more significantly in older ages. So there is an aging of the organism, an aging of the immune system that predisposes us to COVID-19. And just like COVID-19, I could, well, this is because it's just more timely, but there's a number of other age-related diseases I'm sure you're aware of: heart disease, cardiovascular diseases, cancer, neurodegenerative diseases, all of which increase exponentially with age. So clearly we have to do something about aging. And I actually think that COVID-19 is a good opportunity for us to emphasize that point. You know, if people didn't age, or even if we could slow down the process of aging slightly, this would have tremendous impact, including on infectious diseases like the coronavirus. So, I mean, one of the things, if I'm giving a talk, I mean, to a general audience is, you know, I try to pick up an angle of how to convince them of the importance of aging research. So, as Jack was mentioning earlier, I mean, we need to spread the word out of the importance of research on aging. And I think there's different strategies for how we can do that. I think COVID-19 actually plays into it. You know, if we emphasize the health benefits of preserving health and postponing diseases like COVID-19 or any other age-related diseases, I mean, that is one argument we can make. We can say, listen, we're trying to retard aging because we want people to be healthier for longer. You know, we want to postpone diseases. So there will be health benefits. We want people to be healthy. And it's very difficult to argue against health. You know, it's one of the things most people consider sacred. It's not going to be easy to argue against saying, no, I want you to be unhealthy. That's very difficult to argue against. The other argument is to argue in the context of life extension. So if we could extend human longevity, for example, if we could really have a major impact on aging, you know, if we age the way like a Galapagos tortoise does, which is very, very, very slow or even doesn't age at all, then we would see people living over 1,000 years. So that is the other emphasis is on life extension. You know, we slow down or we tackle aging, we therapeutically cure aging, this going to means people living over 1,000 years. That's another argument. I think that's a little harder to defend. I think health benefits, in my experience, tends to be easier to defend. But it is a little bit trial and error, and it also depends on the audiences. I mean, lastly, the other argument is, okay, if we don't do anything, then we'll die. And most of us will die of age-related diseases. The vast majority of people in the world, not just in developed countries, die of age-related diseases. So we need to do something about it. If we don't do anything, then we're just going to have this increased incidence of age-related diseases. So it's another line of argument that I've tried in different contexts. So those are roughly, I mean, there's variations of them. These are roughly the three and combinations, but these are roughly the three arguments I tend to use for emphasizing the importance of aging research. But I'd be interested to know what your perspectives are, and you know, if you have other suggestions or ideas on this. Okay, so now, going back, okay, so we want to, I mean, I'm sure you're convinced we need to target aging and study it and develop therapies. I think one of the big problems we have in the field is that we don't still know why we age. I mean, there's a number of theories. I guess Jack mentioned, you know, DNA damage. You know, that there are theories, there are hypotheses, but we still don't know the precise molecular cellular mechanisms driving the process of aging. That is still not understood. And you know, as a small side note, I have a problem with the Hallmarks of Aging paper because of that, because I think it gives the impression we understand aging when actually we don't. We don't have a conceptual understanding of aging at the same level of cancer, for example. We understand the concept of cancer, mutations, you know, we understand it. We don't understand aging. Again, there are hypotheses, happy to discuss this, but none of them are proven. We don't really know for sure why we age. I mean, that said, there has been a lot of advances in cause for optimists. I mean, one of them I briefly touched upon already are these species that appear not to age, like the Galapagos tortoise. This is painted turtles. This is a particular kind of salamander called the olm. So these are species that appear not to age. That is, you know, in studies in some cases spanning decades, they don't exhibit the exponential increase in mortality. you see in humans or mice or rats or any mammal, actually. You don't observe physiological decline. In some cases, some species, they grow throughout their lives, and they even have, for example, lay more eggs the older they are, which evolutionary, then you can argue, makes sense for them not to age. But the point is that the species appear not to age. Maybe they age out for 500 years. We don't know. I mean, it's hard enough to get PhD students focused on 5-year projects. But we have evidence, at least, that they age much slower than human beings. So at the very least, there are natural mechanisms to slow down the process of aging in ways that doesn't happen in humans. So maybe we can discover what these are and apply those to humans. The other reason for optimism, which I'm sure many of you are aware of, is the fact that the process of aging is plastic. We can manipulate aging in animal models. I mean, Tom Johnson and colleagues identified AGE1. That was the first single gene that had a significant impact on aging. So AGE1 in worms, in this tiny microscopic worms, which you see here, this is a microscope image, a single gene can significantly impact on their longevity. So we can manipulate aging by genetics, by dietary manipulations, which I won't go into detail today, but there's caloric restriction, there's dietary manipulation of aging. We can slow down aging and accelerate it in animal models. And this is a great cause for optimism. I mean, Cynthia Kenyon, who now runs, she's a chief scientific officer of Calico, this Google-backed company that got over $2 billion to develop interventions in aging. That is the rationale behind it, is that we can tackle aging, we can manipulate aging, and let's figure out ways of doing that in humans, because we can do it in model systems. I mean, I've always been fascinated by this, and in fact, one of the projects I started back when I was a PhD student was this human aging genomic resources, this various collections of various databases that, I mean, I don't have time to go into, but I invite you to visit. And one of the main databases we have is this GeneAge database of aging-related genes that really emphasizes and highlights how much advance we've observed in the field. I mean, as a side note, if you're interested in biology of aging, I would also suggest you follow our pages on Facebook and Twitter, as we have I tend to post the latest scientific articles and news on it. So, so just to summarize what is on our database in the latest version, you can see there's over 2,000 genes associated with aging and longevity in model systems. In worms, there's now nearly 900 genes, single genes that extend lifespan by 10 times. That is, a single gene manipulation that will extend lifespan 10-fold. Instead of the animals living a few weeks, they're living months. Even in mice, you can tweak a single gene and have a significant impact. So I'll give you 2 quick examples. These are 2 mice of the same age. The mouse on the right has a mutation in a particular DNA damage repair protein, and so it ages faster, or it appears to age faster than its littermate. So this is a single gene manipulation that appears to accelerate aging in mice. And, it gives the obvious then question is, okay, maybe that's evidence that DNA damage is important in aging. Yes, absolutely. So there is some evidence that DNA damage is important in aging. Conversely, you can slow down age. You can extend lifespan in mice by up to 50% by single gene manipulations. Again, this is an example. These are 2 mice of the same age. The small one, this is very cute dwarf animals. They have a mutation in their growth hormone signaling. So basically, growth hormone, as the name implies, makes you grow. Because they have a mutation in growth hormone signaling, they don't grow as much. However, they live longer. They live significantly longer. They are resistant to cancer. They're resistant to a variety of age-related diseases and degeneration. They don't live forever. They will still age and develop diseases and die, but they live considerably longer in good health, which is what we want if we can apply this to humans. So when you look at humans, okay, so we can manipulate aging in animal models, including in mice. What about humans? I'll show you a couple of examples. So the first one, this was a study by Guerra, which is who you see here, and they focused on small people, as you can see. And the argument was, okay, just like the cute small mouse lives longer, what about small people? So the most interesting result they found was that these individuals are extremely cancer resistant. As you can see, animals with this growth hormone receptor deficiency, they don't die of cancer. So that's quite remarkable, because roughly 20-25% of their relatives that don't have the same mutations die of cancer, which is kind of what you expect in an average population. Their lifespan is not extended, I should say. They seem to have a higher incidence of cardiovascular diseases, for example. So it's not the same impact as in mice of the same mutation, but it shows that at least in one particular age-related disease like cancer, these individuals are very strongly protected, which is quite remarkable that a single gene manipulation can really retard or prevent cancer in these individuals. We also know of an impact, clear impact of the genetics in human longevity. There's been studies in centenarians. They have found relatively few factors in common. So, this is Jean Calment, the oldest human being on record. She ate massive amount of chocolate and port wine from my hometown of Porto in Portugal throughout her life. She smoked. So, she didn't really have a healthy lifestyle. Still, she's the oldest human being on record. So there doesn't seem to be a lot of lifestyle factors in common. It tends to run in the family, however. The best way of you to be a centenarian is to choose your parents and grandparents well. So we know actually from studies, for example, in twins, that longevity has a moderate heritability. There is as a moderate genetic component, but this increases with age. So what it means is that if you want to be a healthy 70 or 80-year-old, you need to watch your lifestyle, watch your diet, don't smoke, etc. But to be really a super centenarian, there are genetic factors to it. Unfortunately, we still don't know what these are. But there is a genetic basis to it. I mean, the point and what I find really fascinating is that clearly there are, we have a very big genome, 3 billion base pairs, 20,000 genes, but very small changes in the genome can have a very significant impact on aging. And that is remarkable. If we can figure that out, we may be able to develop better therapies. We may be able to develop drugs. If we can understand that, we may be able to intervene in aging, because there are these very subtle changes in the genome that can have a very significant impact on aging. And let me give you one example, actually. So, cell senescence, I mean, the limited proliferative ability of human cells. So if you take cells from an individual, if I take cells from my skin, you can grow them on a petri dish, but only for a certain amount of time. Then they stop proliferating and they become senescent. So, for quite a long time, it was thought that the cell senescence process was multifactorial. It was a complex process. There's various factors. Whilst others argue maybe there's a timekeeper, but there's something that just regulates it. And it turns out that yes, there is a timekeeper. It's nothing very complicated. In human fibroblasts, it's actually very simple. It can be a little different in other cell types, but in human fibroblasts, it's the telomeres. You have telomeres that are the tips of chromosomes that they shorten with cell division. Eventually, they become very short, and cells stop proliferating. It's as simple as that. So the point is that complex processes can have simple triggers. And in the context of aging, because we don't really know what causes the process of human aging, we don't know if there may be simple triggers or simple ways of manipulating aging. We know that it is possible to some degree to manipulate aging in animal models, in fact. So with that, I mean, I'll just briefly go through a couple of different projects we've been involved in recent years. So I'm in my lab at University of Liverpool. I run the Integrative Genomics of Aging group, and we combine various experimental and computational methods from machine learning, systems biology, dietary manipulations of aging, which I'm not going to talk about today, cell senescence, evolutionary genomics. I guess one of the drivers of my work has always been that, you have a lot of genes associated with aging, but really the genes have to interact with each other and with the environment. It's not, you can have triggers, but to understand those underlying processes, you need to understand the interactions between genes. So we need to understand the connections between cells, the connections between the components of the cells to understand the process. And then maybe you're able to figure out some pressure points where you can manipulate the process. But you really have to tackle this complexity of biology. I'll show you very quickly a couple of examples. So one analysis we did a couple of years back was this systematic analysis of all aging-related genes, what I call the gerontome, and we just group this into functions. So in an unbiased way, we just look at all of the genes associated with aging in various model systems and which processes or functions they're associated with. And we can also discriminate between pro and anti-longevity effects. So, a pro-longevity would be a gene that if you overexpress it, for example, it increases lifespan. Or an anti-longevity would be like growth hormone receptor, that if you knock out, you have these cute mice that live longer. So you can discriminate between them. So what we found is that, this characterization allows us to cluster, allows us to group genes associated with aging into common pathways and process. Like, for example, in pro-longevity, we see p53 signaling and cell cycle in mice, hypoxia activation and in flies, and autophagy in worms. For anti-longevity, as you'd expect, you have aspects like growth hormone and insulin signaling, which is related. Same with flies and worms, as well as things like oxidative phosphorylation and mTOR pathway. So it allows us to reduce the complexity of the pathways in the study. I mean, we did various analyses in this paper, which we published a few years back, so I won't go into a lot of details, but it's just one more point I would like to mention. So we looked into the conservation of these genes, and we showed that aging-related genes are better conserved evolutionarily than expected by chance. That's fine. But then we wanted to look at the overlap between aging and age-related diseases, because the hypothesis is that age-related diseases derive from overlapping, maybe not the same, but overlapping mechanisms with basic aging processes and mechanisms. So what we found was that indeed we observed some overlap, but it's quite species-specific. So what you see here are four organisms, so mice, flies, and we basically look at genes associated with these diseases, and we look at their overlap with genes associated with aging in the different model systems. And as you can see in mice, there's quite a lot of overlap, quite a lot of diseases that overlap between, at the genetic level, between aging, being pro and anti-longevity, and the disease. There are some in flies, but there's relatively few in C. elegans and very few in yeast. So the effect seems to be species specific. So what it tells me is that, okay, you have some overlapping mechanisms, but probably mice, maybe flies would be the best to investigate those, which is perhaps not too surprising because you have diseases that don't affect really lower model systems. Now, the other aspect of research we've been focusing quite a lot is DNA sequencing. As I'm sure you're aware, there's been this massive amount of breakthrough and technological breakthroughs in DNA sequencing. I mean, this is a photo of me pretending to work with this machine. Whoops. And thanks to all of these technological advances, there's a drop in the cost of genome sequencing. So you can now sequence your genome for less than $1,000, for example, as I'm sure many of you are aware. So there's been a lot of advances in DNA sequencing, and we've been taking advantage of this in our work. So one of them is at the level of transcriptome sequencing, or gene expression profiling. I'll show you one example only. We've published quite a few papers on this, but I'll show you one example from caloric restriction. So we did RNA-seq. We sequenced a transcriptome in rats, in the brains of rats, and exposed or not to caloric restriction. And we find quite a lot of genes overexpressed in caloric restriction, quite a lot of neuroprotective genes, which suggests a protective effect that's of caloric restriction in the brain that's reflected at the molecular level. We did various areas of analysis. I think the main point of this article, which we published some years ago now, so again, I won't go into detail, is that there were a lot of epigenetic regulators, a lot of chromatin-related genes. There were some microRNAs that we looked into, but there seemed to be a strong epigenetic signature although we only looked at gene expression. We didn't have money to look at epigenetics, unfortunately. I applied for a grant to do epigenetics and it was rejected, unfortunately. But that's kind of suggests that there's some epigenetic layer to the regulation of the longevity and health benefits of caloric restriction. Then we've done a number of other functional analyses that I'll mention. So one level of computational analysis I think is quite interesting, and we're still doing this, is at the level of drug repurposing for life extension. So, okay, we have a lot of signatures, for example, of caloric restriction. And I'm sure you're aware there are signatures of drugs. You can download gene expression molecular signatures of drugs online. So this was started with a very simple idea. I mean, can we find drugs that have signatures, molecular signatures similar to caloric restriction? And the answer was yes. We found several candidates, I think 11 or 15, depending on the statistical cutoff we use. 4 of them we tested in worms. 5 of them we tested in worms, 5 of which extended lifespan in worms in a caloric restriction related fashion. In other words, they extend lifespan in normal worms, but not when you put them in caloric restriction. So I think this is a really good example of this application of what I call network pharmacology for drug repurposing. The other application really we're using a lot now is machine learning and artificial intelligence. I mean, we did this, excuse me, we did this review a few years back that I strongly recommend if you're interested in applying machine learning to aging. I mean, the way I usually define machine learning, and so I learned from one of my former postdocs, Daniel Palmer, is it's a bit like, you know, those websites to determine your Game of Thrones character. We'll go there and you answer some questions. Are you male or female? Do you like to kill people? Yes or no? And so on. And then depending on the answers you give, it will match you to the different characters in Game of Thrones. We can do the same thing for pretty much any gene, any protein, any drug. We can look at the features associated with longevity, and then we can look for other drugs or genes. So that is the general concept of machine learning. And we've done that. I'll show you one example looking at drugs modulating longevity. So, so we have a database called the Drug Age Database of drugs associated with aging and longevity. We have over 1,000 entries, over 400 drugs from dozens, well, 27 model organisms. So we have a lot of information on drugs already associated with aging. So then what we did was you look at machine learning applications. We employed machine learning to identify the features, chemical and biological features of drugs associated with life extension. And from there, we build predictive models. So the best models actually employs a combination of biological, that is like gene targets of drugs, and chemical features. And then we apply that to predict new life extending drugs. And we found a number of hits related to drugs that target mitochondria, cancer, and inflammation. So processes you'd expect. I mean, again, I'm skipping a lot of methodological details. I mean, we published this a few years back already. So if you have any specific questions, feel free to let me know. Now, the last approach we've been doing that I'll mention, that Jack briefly mentioned already, is at the level of studying these long-lived species. So one of those species is the naked mole rat, which is, well, which are interested, I mean, not just for their good looks, but also because they can live so long. So these animals can live over 30 years, which is remarkable. They're about a little bigger than a mouse, smaller than a rat, and they're externally cancer resistant. So we sequenced the genome of these animals. It's available online. We did some analysis, but it's mostly a resource for the scientific community. We also sequenced the genome of the bowhead whale. Now, the bowhead whale are the second heaviest animal on Earth after the blue whale. And they've been estimating using these biochemical methods to live over 200 years. I think it's clear that they live longer than human beings. And of course, they live in the wild. So they must have natural mechanisms to protect against diseases that we lack. Like, for example, cancer. I mean, if you think these massive animals, you know, if cancer is just one rogue cell proliferating uncontrollably, they must have natural mechanisms to protect against cancer. So we sequenced the animal, we sequenced the bowhead whale, we also made a genome resource, we did various analyses that I won't have time to go into today. But my point is that we are using these animals as I see them as synergistic, because mice and rats are models of disease. They're models that develop disease, and these are models of disease resistance. So we can find processes, genes, mechanisms that protect against disease, as opposed to mechanisms that cause disease. So I see them as complementary and still very unexplored in biomedical research. The last point I'll mention, in terms of, not research-wise, but more at the level of bringing more people into the field and facilitating discoveries is something I've been involved in, of trying to classify senescence with the World Health Organization. So I published this in Science last year. So the idea is that if we can classify senescence with the World Health Organization, that will facilitate and create a better regulatory environment to do clinical trials for aging. So this is something we're now focusing actually in immunosenescence. If you're interested, Stuart, he's actually affiliated with my lab, and so happy to put you in touch with him, as is something that I think is quite interesting now. It's a long-term project, it will take years, but that I think could have very important benefits for the field. So with that, let me summarize what I've told you. So I told you that aging remains a mystery of biology. Yes, there are a lot of theories, but we don't know for sure what causes the process of aging yet. I told you that our gene age database has over 2,000 genes, which is quite remarkable. And it also means that we need these computational mathematical methods to interrogate data sets, to understand how genes interact. I showed you some of our network analysis to discover new genes and drugs and processes associated with aging. Also machine learning methods, which you can apply to a lot of different processes. Obviously I'm applying them to aging, but you can apply them to lots of different processes. And lastly, I've told you about this long-lived species to find genes that protect against aging. Now, before I finish, just for one quick minute, there's two more points I would like to make. The first one, if you're interested in aging and you're debating someone about aging, people will come up with ethical issues, social implications, what about anti-aging? What about life? There's going to be a lot of overpopulation. So you need to be prepared for that, actually. If you look at my website, there are some, I've actually published an article on ethical implications of life extension some years back. So there are some good arguments against it, but it is something I would suggest you look it up, so you're prepared to argue when you're faced with those questions. I always get questions about overpopulation when I give a public lecture. So it's something that is important to be prepared for. The last thing I'll mention is kind of a different endeavor. So I'm also working with Centaura. I'm their head of science recently, which is a company based in Switzerland that aims to reverse aging via various by radical interventions like gene therapy. So the company is currently recruiting in Europe and also in our lab in Boston. So if you're interested, please check out their website, or feel free to drop me an email. So with that, let me just emphasize, of course, I'm just the spokesperson for the lab. We have a number of talented and ambitious students and postdocs working with us. This photo is a little outdated now. It's obviously before the lockdown. I think it's from last year now. And of course, all of these works I mentioned are made possible thanks to our funders. Reprints and more information about what we do are available on our lab website. And of course, feel free to drop me a line if you have any questions. And I'm happy to take any questions now.